Good afternoon, children. We will complete the chapter, the first part of the chapter, locomotion and movement. Okay. So that finished this this chapter in your human physiology. Now we completed about the skeletal system. Now this is completely muscular system. Now before we begin with the movement in this muscular system, two things. There is actually locomotion. Under the locomotion, you have got skeleton and muscular, two divisions. So to begin with, you want to see in your textbook, if it's an NCERT textbook, he has defined first what is locomotion. Then movement, and then he brought into locomotion. And from the locomotion, he has brought into the muscular movement, and the muscular movement helped by these muscle tissues. As I told you, the one previous chapter we have got cellular organization where you have got complete animal tissue and cockroach type study. Relevance on it. So animal tissue, instead of doing separately, as you do is these systems, particularly nervous system, blood, circulatory system, blood tissue completely we have to learn. And similarly, when it comes to the locomotor, we have already studied the histology of the bone tissue, and now we have to do the histology of the outline of the muscle tissue we'll see, and then we'll go to the striated muscle contraction, the mechanism of the contraction of the striated muscle. That is the important part in this chapter. All right. Now to begin with movement. Now first the movement as it is, you know very well, movement is just moving. Displacement, in a physics language, displacement from one spot to the other. But when you say about the movement, it need not be displacement alone. The simplest movement in your in the living world is the streaming movement of the cytoplasm. Now this streaming movement of the cytoplasm, if particularly in the case of plant cell, so the movement, and then we'll go to the movement. Okay, movement. The movement as it is the simplest. First question, the simplest movement in the living world. Movement is nothing but streaming movement of the protoplasm. Streaming movement, MOVT. This is movement of protoplasm. Protoplasm. Now, you know very well, in the living world, you have got unicellular and multicellular organism. When it comes to unicellular organisms, whether it's animal or plant, what is the main difference between them? The boundary. You know very well, animal cells do not have a thick boundary, whereas the plant cells have got a thick boundary, that is a cell wall cellulose, apart from the plasma membrane. So in the unicellular organism, when the boundary is a simple plasma membrane, the cytoplasm inside shows a streaming movement. So when the streaming movement occurs in the case of plant cell, the whole thing is bound by a hard outer cover. So the cyclic movement is cyclosis. That is the streaming movement of the cytoplasm is seen in the plant cell by the movement of the plastics all along the cytoplasm in a mature plant cell. But the same thing you take an amoeba as a single cellular organism. When the streaming movement occurs, that is what you mean by the amoeboid movement. It will push the cell membrane to one side, and that pushing will form a pseudopodia, a bulge that will help in the locomotion. So streaming movement leads to the amoeboid movement in the unicellular organism amoeba, which has no strict or hard boundary or cell wall. Whereas the same streaming movement of the cytoplasm occurs in the plant cell as well. The cytoplasm, which is confined within the cell wall, will show the cyclosis or cyclic movement. Now, how do they prove the cyclic movement? When you study the organelles in the plant cell, the movement of the plastids could be seen in the cytoplasmic movement. So, the cytoplasmic movement is known as the streaming movement. It is present in the animal cell, the simplest unicellular organism. Whether it's an animal cell or a plant cell, it could be seen. All right? 1 second. Okay. So the streaming movement of the protoplasm is the simplest form of movement. Now, why this is? This is one of the NEET question in the NEET paper in the multiple choice. The simplest form of movement, ciliary movement, flagellar movement, tentacular movement, and movement of the locomotor organ, and the streaming movement. So the simplest in your textbook wording is always you have to be thorough with the NCERT book if you wanted to attempt your NEET. So totally, this is the first question I have fixed up for the regular classes for the test in this chapter. It is the simplest form of the movement in the living world. That's a streaming movement. When it comes to the plant, a streaming mode of the cytoplasm or protoplasm. That is protoplasm. You know very well, protoplasm is nothing but cytoplasm and the nucleus. Cytoplasm is a liquid. Every cell has got a boundary. The liquid cytoplasm, nucleus in the center. When you take the nucleus and cytoplasm along, you call that as a protoplasm. When you bind it to the covering, it will become a cell. Now, this is the first movement of the protoplasm was formed. Apart from the streaming movement, there are locomotor organs. Now, that is cilia, and flagella, okay, and tentacles. Now, where these organisms, the tentacle, cilia is found in the protozoa and paramecium, flagella again in protozoa and euglena, and the tentacles you have got in the coelenteratans, that is in the hydra. So these are all organs which help in the locomotion. But though cilia and flagella is a projection of the cell membrane, now where does the cilia arises from? From the centriole. When the centriole arises, this is the study the structure of the cell organelles. Now, detailed structure of the cell organelles, cilia and flagella. Each cilia have a central microtubule. They have got small microtubules. So they have a skeletal structure in that. Though it's not a bony structure, a hard part. So the hard part in the central core of the cilia and flagella are the two tubes, microtubules, surrounded by tiny, still smaller microtubules in the sets of 7+2 or 9+2. Now, where is the cilia and flagella arises? From the centriole. So, the centriole also has got the same electron microscopic structure of central two tubes surrounded by the sets of microtubules. Three triplets of 7 tubules, 2+7 we call, and 2+9. And that means the cilia and flagella, which you draw like a hair-like structure, it's not a simple hair. It has got a cytoskeleton inside. What is the cytoskeleton? Is nothing but the microtubules. Do you follow? So these microtubules are present in the flagella, cilia, and tentacle. These are nothing but the protrusion of the organelle stretching or protrusion of the cell membrane. Now they are also used as a locomotor organ, movement for the movement. These are, they won't say locomotor organ. It helps in the movement. So whether it is now these organs when they help in the movement, the organs that are helping for the movement need not help only in the movement. The movement as it is, we can say the movement can be of two types. So the movement is known as the locomotion, and movement of curvature. Which they haven't mentioned in your textbook. But what is locomotion? Shifting one position to another position is locomotion. Now, what is movement of curvature? When the main body, this is the main imaginary axis from head to foot. When the part of the body moves away from the main axis, you call that as a movement of curvature. I'm standing, I'm talking, stretching the hand, or stretching my body. My body does not move from place to place, but there is a movement in the part of the body from the main axis. Now, this kind of movement you call that as a movement of curvature. So, what is locomotion? Movement shifting from in its position. That is called locomotion. So, when you talk about the movement, all the movements need not produce locomotion. And see, when I'm moving from here to here, that's locomotion. When I'm moving my hand, it will be a movement of curvature. So movement is a general term used for both the kind of change in the normal position. A body does not move static. It just stuck up to the place. Now, but it moves, it moves, that means what? Either its position is changing or the body is moving from the main axis. So the locomotion is the movement, the part of the movement. So locomotion and movement, you cannot differentiate. All right. So locomotion it's a part of. So we will say all the locomotions will be movement, but all the movement need not be the locomotion. Like this jargon of all the things you could have come. I can say all the alkalis can be, all the bases are alkalis, but all the alkalis are bases, but all the bases need not be alkali. Your chemistry, you know that. So all this all business here also operates. That is, all the locomotion will be movement, but all the movement need not be locomotion. In other words, all the movement of a body need not push your body from your original position to the new position. That is locomotion. So the locomotion and movement can be defined. This is another definition given in your textbook. The statement, they can ask you why. Now the reason, it can be statement question. How can you choose the wrong statement and correct statement? A locomotion is part of movement, or all the locomotions can be moved, all the movement cannot be locomotion. Now the locomotion can move the body from its main axis. So a number of statements they can give you, ask you to choose the correct statement or which is a wrong statement. Statement question can be on that. That's one point. Secondly, so what is this? These are organs of movement. This organs of movement means what? These structures can help in locomotion as well as the movement of curvature. That means what? See, suppose you take cilia. Now the cilia in the case of amoeba or paramecium, lower organisms, it will help to move in the aquatic forms. The cilia are tiny hair-like projections on the surface of the body of the paramecium, and it moves in the water current in the form of a rowing the boat. So it pushes the organ forward. Now you come across the cilia in the case of inner ear, otolith. Otolith membrane, sensory cells have got cilia, and you take the cupula cristae, cupula sensory cells have got cilia. You take organ of corti, you have got sensory cells have got cilia. But the movement of the cilia towards one side, it helps in the reception, either the maintenance sensation, conducting the sensations, or creating an action potential or stimulate the cell. But that movement of the cilia does not move the whole autogonia or ear out of your inner ear. Do you follow that? So the cilia, the organ of movement, can help you in locomotion. At the same time, it can help in the movement of curvature. Now, Paka example, if you take locomotion, the entire body moves. And this can occur mainly in the unicellular structures. Unicellular structure, I am telling, is not unicellular. The unicellular can be an organ, or unicellular can be part of your body. For example, sperm. It's a single cell. It moves. You take this one. What is the thing? RBC. RBC does not. It has no movement. It's got carried away by the water current. You take WBCs, macrophages, or lymphocytes. They show amoeboid movement. They follow. So the ciliary movement, will take the ciliary movement, flagellar movement, and amoeboid movement. So the amoeboid movement. Amoeboid movement means what? Streaming of cytoplasm, streaming movement of the protoplasm is another word you can refer to as an amoeboid movement. So the amoeboid movement, wriggling movement, can help. See, we'll take amoeboid movement. Amoeboid movement. I'm using the word movement. I'm not talking about the locomotion. Now, amoeboid movement, that means what? Putting out pseudopoda with the help of the locomotor organ is pseudopodium. Pseudopodium. This is the locomotor organ. Now, how does pseudopodium help in locomotion? It helps in the movement of the cell like amoeba. It's a unicellular organism. In our body, movement of WBCs or lymphocytes. Lymphocytes. This is movement of for locomotion. But the same pseudopodia, you come to the cells lining the gastrocoelom or coelom of enterocoelom of the hydra, endodermal cells. Now, these endodermal cells exhibit amoeboid movement that trap the particle which comes inside along with the water. So, in the case of hydra, in the endoderm, in the hydra gastrocoelom, gastrocoelom, coelom of the hydra. That's why it's called coelentraton, coelom of the hydra. You have got amoeboid cells. Now, these cells, what are the use of this amoeboid cell? These amoeboid cells helps to capture the tiny particles which come along the water into the mouth, through the mouth of the organism, and it traps that and digests it, cellular digestion. So the amoeboid movement can, in this case, what? It is fixed. It just traps. That means it is helping in the capturing of the food particle, and it is not dislocated. So the amoeba, the pseudopodium, which is a locomotor organ, and that movement of this locomotor organ, you call it amoeboid movement. Okay, the organ is the pseudopodium. Now the pseudopodium helps in the locomotion as well as it helps in the movement of curvature. You follow? Movement, simplest form of movement is streaming movement of the protoplasm. The streaming movement of the protoplasm can occur in an animal cell and a plant cell. If the plant cell is a single free-living organism, like amoeba, it can help in the movement of that. That movement as a whole, we can divide this as locomotion and movement of curvature. These 2 points you must understand. Locomotion means shifting from place to place in your position, and the entire body shifts. Now, this is the one of the characteristic feature of the animal kingdom, mobility. Main difference between the animal kingdom and plant kingdom, the character first is the mobility of the organism. Plants are fixed in their places, animals can move. Now, the move in the sense that a locomotor movement is seen in the animal. You follow? So, this movement of locomotion, movement of curvature means a part of the body moves from its main axis. It's called the movement of curvature, like moving your hand or moving your neck, stretching your body, wriggling your body, or moving the tentacles. See, the hydra fixes a sedentary form, it moves the tentacles. So that is called movement of curvature. At the same time, they follow. So now first you take these movements, amoeba, the organs. The organs which help in the movement are the most primitive one, which is amoeba pseudopodium, next is cilia, then flagella, and tentacles. We'll go from the basic level. Now, so these organs can help in the locomotion as well as in the movement of curvature. So when I define these two, we can say all the movements can produce, all the locomotion will be a movement, but all the movements need not produce locomotion. The movement can produce movement of curvature also. So the next ciliary part, next organ of movement, we take cilia. Now ciliary movement. The movement is called ciliary movement, and the organ is cilia. Cilia is nothing but formed from the centriole, structure of centriole, and it has got cytoskeleton. Is not a single hair-like structure as we draw. In electron microscopy structure, each cilia is a set of microtubules. That is called cytoskeleton. The microtubules are larger tubules in the center, surrounded by tiny sets of microtubules. We call them as 9+2 and 9+7. 7 sets around in the case of flagella, 9 sets around in the case of the cilia. So the whole thing is a set of tubes. Now these tubes are called microtubules. They are called the cytoskeletons. Where do they start from? Centriole. So the centriole which we draw like small dots, they are not a solid dot, they are nothing but a group of tiny tubules. Suppose you take central, you have got two long tubes you are keeping like this, purely diagrammatic, okay? Now surrounding that, and on each one is a tubule, a number of tiny tubules. Here, then again around. So the cross section, what will be in your textbook? You have got two central tubules, and this is a central one, surrounded by these tiny tubules. Now these are connected with one another. This has got a covering. Structure of the cilia you should study. You could have studied in the cell organelle. The whole thing is a cilia. Like this, you have got triplets connected central one. So now this is connected to the membrane as well as they are connected with one another. These are connected with one another. The electron microscopic structure of cilia and flagella, you should know that. Botany, you could have studied cell as a unit of life, cell and cell organelles in the first volume only, the second unit. No, so these are these tubules, these tubules. Tubules means smaller ones are called tube. Little bit bigger one we call that as a tube. Now they are called the cytoskeletons. Cytoskeletons. Now these cytoskeleton, these tubules are present in the centriole also. Centriole is not a small. How will you draw centriole? If you take a nucleus cell, nucleus is here. You have got the two centriole. Now this cytoplasm around that is known as the centrosome. Now what is the function of centriole? During the cell division, the centrioles extend from one end to the other, form the astral fibers, nuclear spindle. So these fibers are nothing but the tubules extending. You follow that? So these centrioles, actually the cilia is not a blind structure or a mass of cell. It has got a cytoskeleton. Okay. Now we are only the movement. So cilia produces ciliary movement. So what are the movements exhibited? Flagellar movement, ciliary movement, pseudopodial movement, or streaming movement of the cytoplasm, locomotor organ, amoeboid movement, or locomotor organ is pseudopodia. Ciliary movement. Locomotor organ is cilia. What is cilia? It's set of microtubules with a central larger tubule surrounded by the tinier tubules of sets. So same structure for flagella. So when the centriole protrudes in the form of cilia, the centriole is, you don't call that as a centriole, you call that as a basal body. Basal body is nothing but a centriole when it extends outward to the cell in the form of a tube, long microtubule. That thin microscopic tube only we call that as a hair-like structure called cilia. But if you look under the electron microscope, the hair is not a single rod, it is nothing but a set of tubes, tiny tubes put together. Do you understand? So the cilia starts from centriole only. When the cilia arises in the centriole, you call the centriole as a basal body. If there is no protrusion outside, it remains as a centriole and gets involved in the cell division, forming the astral rays and the nucleus spindle. All right. Now, ciliary movement. Now, ciliary. Now, what are we bothered about now? What is the movement produced by cilia? It's a locomotive organ of movement. So this can produce locomotion. It can produce locomotion. Let's see. Go forward. Locomotion as well as it can produce movement of curvature. Movement of curvature. Okay. Now locomotion. In the case of locomotion, take for a paka example, Paramecium. Spelling Moecium. Paramecium is a protozoan unit. It's called slipper animalcule. It has got a number of cilia around the body. The cilia hair moves like a O, and that propels the body again. Now, what about the movement of curvature? For example, you can take your inner ear. Movement of the hair cells of the organ of Cotton, hair cells on the otolith membrane, hair cells on the ampulla of the cristae. Very easier one, but one of the questions in the multiple choice they can ask you, the ciliary movements or the ciliary movement of curvature or cilia helps in the ciliary movement is seen in your body, in the organ, inside the organ, such as in the fallopian tube. Fallopian tube is a place. Fallopian tube. We can see it many places. See, olfactory epithelium, cilia, trapping the dust particle. Trachea, cilia, trapping the dust particle in the air which passes through. Okay, and then you take the stomach, inner lining of the stomach. There are ciliary movement is there. The cells have got cilia and helping in the paralysis and the rugae. So the ciliary cilia occurs in the inner mucous membrane cells. Now these cells, the movement of the cilia helps in the movement of in some function it has got according to the particular area. If it is the inner ear, producing the action potential, receiving the stimuli, and if stimulated. And if you take the trachea nasal epithelium, trapping the dust particle. If you take in the fallopian tube, the ciliary cells, the fallopian tube cells pushes the movement of the ova back towards the uterus. If you take the stomach, it helps in the movement. It helps in the peristaltic movement. So all these movements where the cells have fixed the cilia, the movement of curvature. It's actually the cells do not move. The cilia is not stiff like this, so it moves. That's what you mean by ciliary movement. So the organ which are helping in the locomotion can help in the movement of curvature also. Alright, now the next one you take the flagellar movement. Now flagellar movement is flagella's organ. It helps in this what? In Euglena, the protozoans, complete propulsion. Locomotion. Now locomotion in the protozoans. Locomotion in protozoa. Now in our body. Now help in this particularly the sperm, spermatozoa, sperm, the movement of the sperm, flagellar movement of the sperm from cervix up till reach the ovum for fertilization. So the movement is the flagellar movement is exhibited in the sperm in our body, a single cell. Sperm also is a single cell. So single cell gets located. Now other type of flagellar movement in a sense, the flagellar movement, particularly for locomotion, if you take the movement of the flagellum, it's mostly unicellular structure like protozoans. Now Euglena, the species of Euglena. In the case of Chlamydomonas, take Chlamydomonas. Now Chlamydomonas moves with the cilia and not with the flagella. Now particularly Euglena, the two flagella, one will be short, one will be long. So both the flagellar movement is exhibited in the protozoans. Now you take the tentacles. Now tentacle will take the next part is your movement of curvature. If we take movement of curvature, the flagellar movements helps. Flagellar movement is not particularly in the cells, a particular area of the cells. It does not help. It doesn't involve much the inner organs of our body. Our body flagellar movement is exhibited mainly by the sperm. Now you won't find a single cell with a flagella in your body moving inside the locomotion. And when you talk about the cells lining, it will be more ciliated cell. You won't find the cells of your mucous lining layer or any organ of your body. You've got a flagellar cells over there. So now when you talk about the tentacles, now tentacle won't involve in our body in the lower organism. Now the tentacles. Now why are tentacles? Okay. Now, why this is, the movement of, this is locomotion. At the same time, the flagella can help the organism to capture the prey from the surrounding. How does it? When Euglena moves, it will help the Euglena to move the lashing of the flagella. At the same time, when it moves around, the water current is pushed. So the flagellar movement, fixed movement, if it is propelling movement, it's a locomotion. When it is a private, it's an aquatic form. It won't be stand still in the water. So when it is moving in the water currents, lashing it in the water current, it will push the water current towards the organ where it can ingest the water along with the tiny particles that helps in the nutrition. Here, locomotion, when it comes the capture of the prey, that's a movement of curvature. The flagella, this animal does not move. It can, when it moves, actually, the water is pushed into it, and it helps in the case of nutrition. Same manner when you take the tentacles glaringly will come in the cnidarian, coelenterate form. These are protozoans. Next group, when you come to the coelenterata, is very famous for the tentacles, you know that. Now, even the ciliary movement, the flagellar movement, another one, you know, where now if you take the sponges, you've got canal system. The canals. In the canal, water, there are tiny pores through which the canal forms and goes the spongiosole in the center, and the space through the canal lined by the cells. Now they keep the current moving in the canal. Towards the central, that is the spongiosole. From the spongiosole, the water is pushed out to the exterior. Okay, so now again in sponges, it maintains the flow of liquid through the channel. So in that case, it's not locomotion, it's a movement of the cell. It helps in the movement of the liquid is one of the curvature. You won't say the body of the organism moves, it helps in the other part, propelling the water, capturing the prey. Now, take the tentacle. Tentacle, particularly if it is in the case of tentacles, we don't have tentacles in any cell inside the body. But the tentacles are a characteristic of the group Cnidaria. Cnidaria. When I say about the Cnidaria, you should know about you remember the example Hydra. Now, Cnidarians are nothing but coelentratans. They are called Cnidaria because they have got the stinging cells, cnidoblast. Now, all the group of these animals are also known as coelentraton because their body cavity acts like a digestive system. Paka example is Hydra. If we take the Hydra, the tentacles help in the locomotion of the Hydra. Now, how the Hydra moves? Looping movement or somersaulting movement. The tentacles help the animal to move, particularly locomotion. And capturing the prey, another part is what? It remains there. The moment that the tentacles pushes the water back and the water current goes inside, and then the cells kill the prey, helps in the capturing the prey. So the tentacles can be used for the locomotion as well as for the movement of curvature in capturing the prey. So these are how these organs, what do you understand from this? Now all these organs of movement need not help only in the locomotion, they can do other function also. Other functions of what? Capturing the prey or keeping the water current moving through the body, as in the case of spongiosole. So the organs which are used for movement, they need not do only locomotion work. They can be used for other purposes also. In the case of cilia, not only locomotion. If you study the structure of Paramecium, you have a small groove that's called cytopharynx. All the cytopharynx are lined by cilia, so it helps the water to move inside, attract the particle which is entering inside. That's the food of the animal. So all these organs of movement helps in locomotion as well as in the other functions. You call that other functions in the body moves to capture the other function. You call this a movement of curvature. So that is about organ of movement, particularly cilia, flagella, tentacles. Now when you go to the human body, now our body cells show three kinds of movement. Ciliary movement and flagellar movement where the sperm, that's the only human body we can make out. Amoeboid movement, that's pseudopodia amoeboid movement, ciliary movement, muscular movement. So the amoeboid movement, this is another question. Why am I explaining all this? This is a fun introduction of your lesson. But introduction of the lesson, these movements are there. That can be asked for a multiple choice. They are only bothered about the NEET point of view. So the multiple choice, they can ask you simplest form of the movement over. And the organs of movement, they can fix. So organs of movement can be, it is a skeletal structure, it won't come there. Muscular movement only, a skeleton will come. Organs of movement can be cilia, flagella, or tentacle. Okay, now that is the locomotor organs. Locomotor, another thing, they can ask you locomotor organ of protozoans. They can give you muscle and the bone, cilia, or flagella, or multiple choice you can. Particularly statement question they can ask in this part. All these there in the beginning part of your textbook. The same point is explained in a different manner, that's it. But the particular statements you should be thorough. That is, all locomotions can be movement, but all the movement need not be locomotion, that's one. And the organs of movement help not only in the locomotion, they can do the other functions also, like capturing the prey or defending the body or keeping the current mover. Then another important question, multiple choice, ciliary movement is seen in, and you can give your body. And only in our body, we won't give you stomach easily. They can give you ciliary movement is seen in the cells of in the fallopian tube. Or to muddle up, if I have to set the paper, I will say in the otolith membrane. Or I'll blindly say inner ear. You won't think of the inner ear when I say ciliary movement. Because your sensory cells are ciliary cells. And the movement of these cells only creates a action potential. So I will say internal ear, or organ of Cortine, or macula, cristae, fallopian tube, and stomach. There are three places particularly given in your textbook: trachea, fallopian tube, stomach. Stomach is not given, fallopian tube and trachea is given. But you should know when you study the other organs, wherever the cilia is there and trachea is there, at the same time you have got the nasal epithelium, your hair cells are there, sensory cells will be with cilia, and inner ear you have got. So these parts, you should remember which are the parts the cilia is present, what is their function. All right. So then next question. This introduction will appear very simple, but they can fix a NEET questions in the first part. Statement question is very common. Or they can give you one set of organism on one side and the other set of locomotor organ. Now I can give you sperm, human sperm, spermatozoan. I won't say sperm, spermatozoan. Then paramecium, amoeba, hydra. I'll give the organs of separately. When tentacles I can give you, cilia I can give you. Now, when I give the sperm, I won't give you another organism which can move the flagella. Flagella will be the sperm, amoeba, amoeboid movement. So, these organs will be there. You should match the following. You can do that. So, these points you should know very clearly. Alright? Now, the ciliary movement, if I say sponges, is there? The movement, the flagella, the movement lining the canal of the sponges keeps the water current moving across. Now, ciliary movement, if I say paramecium, you will fix it up. In the cytopharynx of paramecium, I will say. In the cytopharynx means a groove like your throat. The paramecium, if you have studied, you could have studied, the paramecium structure is like a sole of a slipper. It's called slipper animalcule. At the side, you have a groove. This is called cytopharynx. And the end only is the mouth. You know, cytoplasm, and it has got two nucleus and the cytoplasm, all etc. Now, we are only bothered about this. Now, here, this area. See, if suppose this is a cytopharynx, this part alone I'm saying, the mouth is here. Now this is the cytoplasm. Now the mouth is here. All over here there is cilia. So the water current comes through that, and this cilia pushes the water current forward, and the particles are trapped, and the particles enter in. They form the food vacuole. That is how you should know the digestion, how digestion occurs in the case of the paramecium. Simpler ones. So why did I waste your time for the front part of your lesson? You can get a statement question, multiple choice, because all these organs are mentioned over there. Some naming also is there. Now, the ciliary movement, particularly your body, you should know. Don't forget fallopian tube, stomach, trachea, nasal epithelium, inner ear. Now, here nasal epithelium is not mentioned in their book. But you remember that can be also an option for you. Right? Now the second part, now we are going to the movement in the muscular movement. In the cells of the human body, the cells of the human body exhibit three types of movement. The body cells exhibit three types of movement. One is ciliary. Now, first we say amoeboid movement. Amoeboid, flagellar, and muscular. Our body cells, the cells of the body, amoeboid movement, and flagellar movement. Only these three as mentioned: flagellar movement and muscular. Now, why he has given this muscular? There is no ciliated cell in your body which moves from one place to another. When you talk about the amoeba, Pakka, you have got the WBCs, all the blood cells. And WBCs, leukocytes, whether the A granulocytes, whether leukocyte, monocyte, or macrophages, all these cells are capable of amoeboid movement. Because of the amoeboid movement only, the WBCs can penetrate through your capillary wall and can come out of the capillary wall. That is called diapedesis. But whereas RBCs cannot come out of the blood capillary. They have no amoeboid. They cannot put out pseudopodia. RBCs are carried away by the blood current. They cannot move against the blood current. But WBCs, particularly macrophages and lymphocytes, now they can swim against your blood current and they can come out of the capillaries and then pass through the body tissue, body fluid, and reach the place where there is an injury. That's why they are called soldiers of the body. Where they reach the injury, the cut end, where the microbes are entering from the exterior, they fight it off and they engulf them. They engulf them with the help of the same manner like amoeba. So these cells, the amoeboid movement is peculiar to our cells of lymphocytes and macrophages. In general, we can know it is a WBC. Right? Now, then flagellar movement, I told you already, the one characteristic one is your sperm or spermatozoa. Now, muscular movement is the common movement which helps in the entire body, the locomotion of the body as well as movement of curvature. In many angles, you move your head. You move your head or a hand or a finger or any part of your body. That's a movement of curvature. Stretch your body, that is your muscular movement. Now, the same muscular movement helps you to move, run or move or swim or all the activities which you carry out. So, that is the locomotion. Right? So the movement, the locomotion particularly, varies from animal to animal. That is, shift. Locomotion means what? Moving from place to place. It will vary from individual to individual according to the habitat where they live, according to the situation and surrounding. So every time I won't be running on the road, then I'll be mad. So yeah, that's why when you're playing, you cannot be walking leisurely and strolling around. So depending on the situation where you need how to move. Another thing, it depends on the environment, the habitat where you live. Do you follow? So the muscular movement is the one we are going to study now here. So the muscular movement involves The muscular movement involves three coordination between three systems in your body. That is the muscular system, and second nervous system, and skeleton. Now these three systems should coordinate one another to form the muscular system of a movement of your body. That in our body, you have got the three movements exhibited. Most important is the muscular movement, and the muscular movement involves under the control of muscular system, the different muscles, and particularly the muscles when they are involved in the locomotion. Say you take peristalsis. Peristaltic movement is the beating of the heart is also motion. It's also a movement. So the peristaltic movement, the propelling movement of the stomach or the intestine, pushing the thing forward, peristalsis, that is also movement. When the food is passing the gullet to the stomach in the form of small roll, deglutition, that is also movement. And when your undigested matter moves along the large intestine, it's called pendular movement. Small jumps, that's how form the fecal matter. So these are the movements involved inside the organ without the location of the particular stomach or wherever it occurs or a gullet. They follow, but these movements, so the movements in your body, Pakka locomotor movement is the one which move your body shifts in any direction, whether it is a movement of curvature or either body moves. If I move my hand also, I need the bones are involved and the stimulus is involved, nervous system is involved, and muscles attached to the bones are involved. So the muscular movement particularly, the all movements are muscular, nervous and skeletal coordination, and the muscles are under for this. The muscles are a whole in your body. You know very well skeletal muscle and smooth muscle. I should not use the word smooth. Say I use visceral muscles, skeletal, visceral, and cardiac. Skeletal, visceral, and cardiac muscle. Now, the skeletal muscle, as you know, they are attached to the skeleton and they bring about the movement of the. So they are called skeletal muscle because they are attached to the skeleton. And visceral muscles are present in the inner organ. Now, they are called. This is also known as striated muscle. They show characteristic striation, and this is called smooth muscle. And this is the one in between the two. It has striations, dull striations, but it has dull striation. But you can't call that as a striated muscle. But neither can we call it a smooth muscle. It's not completely absent. It's intermediate. So it is a dull striations are there. Dull striated. So I cannot use the word smooth. But what happened? This is voluntary under your willpower, under your nervous system completely. Unless the nervous system stimulates, this muscle won't contract. So this muscle won't contract. The muscle has to be attached to the bone to bring about the motion. That's what we mean by the joint. Now here you call it as a involuntary. It is not under your control. Whether you like it or not, the food you which you eat will automatically just move from your stomach to the part. Otherwise, it's indigestion. Something is wrong. Your stomach will get bloated. You will feel uncomfortable. And similarly, when you take about the cardiac muscle, involuntary. What is involuntary? Whether you like it or not, one day it will stop and one day you can't tell the heart to work and then stop on and off. So these are these things. So the cardiac muscle is similar to smooth muscle in its action, but similar to striated muscle in its structure. So these three muscles are in your body, major muscles. Now, before we have to learn completely about the skeletal muscle structure, before that we'll go to smooth muscle and the cardiac muscle, referring to your first chapter, histology. How do they occur, right? Now, for cardiac, smooth muscle. Smooth muscles, they are not in bundles. It, of course, it's a tissue made up of cells, a number of cells. Now, these cells of the smooth muscles are I hope my writing. Okay, try to write better. The cells of these are spindle shaped. Spindle shaped. Spindle shaped. That is, they have got a tapering end. Anything is spindle shaped, a tapering end, a broad center, and a tapering end. And they are the cytoplasm. And the full of cytoplasm. There is a nucleus and full of cytoplasm around. These are the cytoplasm. Now, in that, there are small fiber protein filaments called myofibrils. The myofibrils are scattered here and there. There are small myofibrils. Now, these lines are myofibrils. So, spindle shape, this is the nucleus of the cell. Now, this is the cytoplasm of the cell. Now, this is the myofibrils. These are called myofibrils. Myofibrils. Now, these myofibrils all have got special proteins, the contractile proteins. You call actin and myosin, tropomyosin, troponin and tropomyosin. That is myosin and actin are the contractile proteins. Now, these proteins make up these myofibrils. So, such myofibrils are few, not many, but they are not arranged in a particular pattern. They are scattered in the cytoplasm. Alright, now because these cells are spindle in shape, they are arranged in the form of layers. That is, they will fit into the shape like this. Suppose one cell is here, and another one will fit in here. Another one will be like this, and another one will come over here. So they form layer by layer like this. So they are in layers. Now these layers can be lengthwise or circular. So when they are arranged in the lengthwise form, all the cells are like this, lengthwise. Now then you call that as a longitudinal smooth muscle. So the cells are arranged layer by layer, but they are lengthwise. Now this is called longitudinal muscle. Now when they are arranged in circular form, suppose you have got a circlet of cells. One cell here, another cell layer, another layer here cells. And they are in the circular manner. This is called circular cell. Circular. They won't say circular smooth muscle, circular muscle, that's all. And this they will say longitudinal muscle. Longitudinal muscle. There is no longitudinal muscle or circular muscle in your body. Skeletal muscle, smooth muscle. The smooth muscles are when they are spindle shaped and they have a central nucleus. Cytoplasm empty, ends are tapering, and the cytoplasm there are a few myofibrils. These myofibrils are only responsible for the contraction of these cells. The cells are arranged in the form of sheets. They are in layers. They are in arrangement of the cells are in the layers. Now they form a sheet. So these layers are kept lengthwise. It's called longitudinal smooth muscle. When they are circular, it's called circular smooth muscle. Now this is circularly a circular smooth muscle. Circular smooth muscle. Now this is what first chapter. The wall of the digestive tract has got the 4 layers. This is one of the major NEET questions. Is there the diagram question? They'll give the diagram, label it. It's a famous question. The wall of the entire gut has got 4 layers. Outer serosa, then muscular layer. The muscular layer, they say outer longitudinal, inner circular. What is this muscular layer? They are all visceral organs. They are smooth muscle. So smooth muscle kept longitudinally and circularly. There is no longitudinal muscle, separate circular muscle in your body. In our organ, the muscles are arranged lengthwise or circular. Now this is the arrangement of these smooth muscles. Now these contract, when the longitudinal muscle contracts, what will happen? The length of the organ will shrink. When the circular muscle contracts, now the girth will shrink. That is, the width will decrease when the circular muscle is contracting. Say circular muscle contracting, the gap will become narrow. When the longitudinal muscle contracting, the length will decrease. That's all. This produces the movement, propelling movement in your inner organ particularly. So the smooth muscles are the shape of the smooth muscles. Now compare this with the cardiac muscle. When you take the cardiac muscle occurs only in the heart. Now smooth muscle occurs in the viscera, in all the viscera. Viscera means inner organ. So they are known as the visceral muscle as well. Now, viscera means inner organ. All the smooth muscles occur in the inner organ of your body. So another name we can say visceral muscle. Now, in the case of cardiac muscle, it occurs only in the heart. But each cell of a cardiac muscle is a fiber, a long thread-like structure. But now each one is a long thread. Okay. Now the thread has got a number of branches, not a simple thread like this. Okay, this is a cardiac fiber. In the cardiac fiber, you have got cytoplasm. The plain thing, and the blue thing within the line is considered the cytoplasm. Now, here myofibrils, there is no central nucleus, one single nucleus. You have got many nuclei scattered all over. Nuclei are not set, many nuclei scattered everywhere. And you don't find characteristic myofibrils here and there, that's it. Then the striations in between here and there, you will have simple dull striations. These are stripes. There's no regular. Now these are striations. Now the nucleus. Oh, wait, 1 minute. I'll just put the nucleus round so you can make out the difference. Each filament is a cylindrical structure with the branches. Now like this, okay, and the nucleus are all scattered all over. And there are dull striations. Striations are very dull and irregular, like this. And this branch will connect to the next cell. The next cell will be connected to the following one, like that. So this is the next cell. There is a gap here. Now, diagrammatically, I'm showing so much of a gap. What will happen is a single thread, a microscopic structure. So they can stick on together well. The cell membrane, these two cell membrane will opposing each other. When they oppose each other, this area will get thickened. Now, this area is known as the plasmodesmata. No, not plasmodesmata. They are known as intercalary disc. They are called intercalary disc. Intercalary disc. Okay. So these intercalary, that means what? The cells are connected to one another to the plasma membrane. At the same time, the cells are not separate; they form a network. Just like a neuron. Neuron forms a network with the help of the dendron and the axon. That is called synapses. So similarly here, the cells are irregular and they have got the connections with one another. They form a network. As a result, the conduction can spread from one cell to the other. The main idea of the network is what? The conduction can spread. When you study the cardiovascular system, SA node, action potential started in SA node, it spreads all over, radiating. It passes through from SA node to the AV bundle, and bundle of this Purkinje fiber. So the conduction or the stimulus can easily thread through the whole organ. So the entire organ can function together. This is a special organ, that is your heart. So the heart muscles or the special muscle and the cardiac fiber is a branch, single cylinder. Each cell is a cylindrical structure. It has got processes. It has got many nuclei scattered and dull striations. And the cells are close when they oppose each other. The cell membrane of both the things get thickened. That is called intercalary. What is the use of the intercalary disc? It acts like a booster. The intensity of the stimuli, from when it passes from one cell to the other, the strength of the stimuli does not decrease. So the intercalary, it is boosting up maintaining the strength of the stimuli continuously, still it spreads through the entire organ. Now compare this to your voluntary muscle. We will compare this to the voluntary muscle. Now skeletal muscle. Now we are coming to your actual part, which is skeletal muscle. Now the skeletal muscle muscle. Okay, skeletal muscles, if you take a bundle of muscles, this is a bundle. Okay, and it has got a number of, now this is the entire thing is the bulk of the muscle. The bulk of the muscle has got a number of bundles. Each bundle is known as a fascicle. Now this is a fascicle, there is one bundle. Here I am cutting. I cut it, this diagram is there in a book. So they will ask you, each one is made up of a number of bundles. Each bundle is known as fascicle. It is inside. Now each bundle is known as the muscle fascicle. CICLES, fascicles. Now these muscle fascicles are connected in between, they are connected by connective tissue. The connective tissue in between them is known as fascia. Fascia is a connective tissue which is in between the fascicle. This is the fascia. They cannot be just kept loosely. Fascia. Now you take a single fascicle. Now the single fascicle in turn is made up of a number of muscle fiber. Now each muscle fiber is a cell. Now this is the muscle fiber. This is a bundle is the fascicle. I am taking a single fascicle alone. A single fascicle, I'll draw the muscle fiber smaller than that. That will be better. Now this is the bundle, the entire bundle. Okay. The entire bundle. Okay. Now the inside, in the bundle, you have got now I have got a number of, this is called the fascicle. These rounds are called the fascicle. Okay, and in between the gap is filled with the fascia. Now you take this fascicle out separately. Now each fascicle in turn, each bundle in turn has got, this is a fascicle. Inside each fascicle, you have got a number of fiber. Now these are the fibers inside. These are the fibers, muscle fiber. Now each muscle fiber is a cell. So inside the fascicle, you have got the muscle fiber, many muscle fiber. Understand? Now, see, this is one fascicle, there is another fascicle, all this put together in one big bundle. Then the fascicle has each fascicle has got a number of fibers. These are all muscle fiber. The entire thing is a fascicle. The entire thing is a muscle bulk. The entire thing is a muscle bulk. Okay, muscle bulk. Now each bundle, this round is a fascicle. Now each fascicle has got a number of muscle cells. Now these cells are known as fibers. Alright? So a single muscle cell you have to take. Understood that? So the single muscle cell if you take, now you take this. Now this is a muscle cell. Muscle cell is a long thin fiber. It's a fiber, so it has got a plasma membrane, outer covering. Now this outer covering is known as the sarcolemma. So inside that, now inside you have got now it has got many nuclei. Automatically in interior, this blue thing interior is completely sarcoplasm, cytoplasm, it's a cell. This is like a cell membrane. So inner what will be there fully? The inner part is completely about the cytoplasm. This is the cytoplasm. Whole thing is filled with the cytoplasm. Alright? It is filled with the cytoplasm here. Okay. Now this is cytoplasm. In the cytoplasm, you have got a number of now in the cytoplasm. Now it has got many nuclei. The nuclei are many, and the nuclei are distributed on the now nuclei are distributed. Suppose you take a fiber, I'm cutting it across lengthwise and showing the interior. Outer covering is the plasma membrane. The inner one is the cytoplasm. And you have got many nuclei. And the nuclei are arranged in the cytoplasm in a row by row like this. Now because it's a section, like this. Say my finger is a fiber, the skin is the outer covering. The peel of the skin, there will be muscle tissue will be there. Now the muscle is like a cytoplasm. And on top, between the muscle and the skin, you have got a number of nuclei, row by row by row by row. So multinucleated, the nuclei are arranged in a straight row. I am only showing a single straight row. In the cytoplasm, you have got a number of special protein, segments of protein fibers, filaments. Now these filaments are called myofilaments. And the blood supply, the blood vessels will be running beneath the cell membrane. So closer to that, there's a blood membrane, blood vessels, capillaries. So each cell has got a covering that is sarcolemma, and the cytoplasm known as sarcoplasm. It has got many nuclei, and the nuclei, I have taken a section. Don't imagine there will be only one row, just like this. The skin is a cover, muscle is a cytoplasm. In between the skin and the muscle, the rows and rows, the cytoplasm only the nuclei will be there, but the nuclei are arranged in a particular pattern, row. So the central part, you have got something arrangement is there. Now the blood vessels are close to the outer membrane and the diffusion occurs through that. So from the cell membrane, what is in the cytoplasm? The cytoplasm has got special structures called myofibril proteins. Now these proteins are called myofibril, thin finer threads. This is just a thread, small plate-like structure of myofibrils there. The myofibrils all over the cytoplasm, you have got the myofibrils. In the cytoplasm, there are a number of myofibrils. These are the myofibrils run throughout. In the cytoplasm, you have got a number of myofibrils. Okay, make out that? Now, let's come to the labeling part. Now, this is a single muscle cell. The muscle cell is known as muscle fiber. The muscle fiber is a cell. Right. Muscle cell. The muscle cell is called muscle fiber. Fiber because a thread. It has got outer covering is called sarcoplasma. Now the central one, the yellow thing is the sarcoplasm. Sarcoplasm. And it has got many nuclei. The green thing is a nuclei. Many nuclei. Many nuclei. Then the blood vessels. On top blood vessel. Then in the cytoplasm, the pinkish lines which I have shown, they are called myofibril. So don't mix up the word myofiber and myofibril. Each cell is called myofiber or muscle fiber, and each the inside the cytoplasm of the muscle fiber have got thin filaments, thin thread-like structures are scattered in that. These thin thread-like structures are arranged in a parallel manner, a longitudinal manner, and that are called myofibril. You follow that? Now you take each back again. Bundle of muscle bundle. Muscle bundle has got a number of bundles of fascicle. Now, there's a muscle made up of fascicles. Yes. Each bundle is known as fascicle. Fascicles. Now, each fascicle has got the muscle cell. Muscle cells are called myofibers. Myofiber is different from myofibrils. Myofiber or muscle fiber. Now each muscle fiber is a single cell. The muscle fiber has got a plasma membrane that's called sarcolemma. It has got a cytoplasm that's called sarcoplasm. In the cytoplasm, characteristically, there is an endoplasmic reticulum is plenty. So the endoplasmic reticulum is known as a sarcoplasmic reticulum. Anything connected to the muscle is sarco. So the characteristic feature is the sarcoplasmic reticulum. Sarcoplasmic reticulum. Reticulum means what? Network. So the endoplasmic reticulum in the muscle is called sarcoplasmic reticulum. In the muscle, this endoplasmic reticulum is very important. Go to the cell. What is the endoplasmic reticulum? There are two types of endoplasmic reticulum: smooth endoplasmic reticulum and rough endoplasmic reticulum. Now, smooth endoplasmic reticulum helps in the synthesis of the lipids, and the rough endoplasmic reticulum involved in the protein synthesis. Rough endoplasmic reticulum is called rough because ribosomes are stuck up. They are minute channels. In the case of muscle cell, the sarcoplasmic reticulum is very important because it mainly helps in the conduction of the calcium ion, which actually which is produced by when the muscle is stimulated. The action potential release of the calcium ion is stored in the endoplasmic reticulum and it is released from the endoplasmic reticulum when the muscle receives the stimuli from the nerve ending. Now, when the stimuli will be received when the nerve is stimulated, the nerve will be touching the muscle fiber. So each muscle fiber will be in touch with the axon, a nerve fiber, a neuron, end of the neuron. So each muscle fiber will be, suppose there is a neuron here, Now the neuron, the end of the neuron is known as the axon. The axon will be touching each muscle fiber will be touched with the axon. Fused with that. So this place where it is fused with each other is known as the motor end plate. So a motor unit is a muscle fiber. Now this is the axon. Axon touching the sarcolemma of the outer membrane of the muscle fiber. When this place where you have got the axon plus the plasma membrane of the muscle fiber, that place when they attach it to one another, the thickening is called motor end plate. Now that forms a motor unit of the muscle structure. Back again, functional unit of brain, functional unit of, see, this is a motor unit, it's not functional unit, it is given as motor unit. It forms a motor unit where when the stimuli comes from the axon, the end of the axon will have neurotransmitters. Now, if you remember the motor end plate, you have got the conduction of the nerve impulse, axon ends up like this, and this is the muscle fiber. Now, the axon has got neurotransmitter. Now, they produces acetylcholine. Now, these acetylcholine. Now, this thickened area only the motor end plate. Now, this is the motor unit. This part only is a motor unit. That is the axon. This is the sarcolemma, the plasma membrane of the cell, PM of the cell, or sarcolemma. I should not use the word PM because it's, the covering is known as the sarcolemma. I'll write SM, sarcolemma, and the fusion, this plate is the motor end plate. Now this area is the motor end plate. Motor end plate. Now this is the end of the axon. These are the neurotransmitters, the cycles, and this is the muscle fiber, and this area is the motor end plate. Now this is the acetylcholine. Acetylcholine will be liberated by these fascicles. Acetylcholine is filled here. Through that it passes into touches the plasma membrane. Now this is the sarcolemma. Protoplasm of the muscle fiber. Now this is the axon. There is a motor end plate. This place is the motor end plate. Motor end plate. See, now this sarcolemma, the axon, end of the neuron, that's the axon, will touch the sarcolemma, fuse the sarcolemma. The fused area is known as the motor end plate. So inside the axon, cytoplasm is there, and there will be small vesicles will form. These vesicles will have neurotransmitters. What are the neurotransmitters? Chemicals which are produced. What are the two neurotransmitters? Adrenaline and acetylcholine. So here acetylcholine will be produced. So the acetylcholine forms in small vesicles, and the vesicles come to the end and burst, and the chemical passes through the fused area through the membrane into the cytoplasm. Now, how does it enter into the cytoplasm? The cell membrane for tiny tubules called T tubules. Now, T tubules will join with the endoplasmic reticulum. So the neurotransmitters first stimulates the motor end plate, thus action potential is. And these acetylcholine will pass through the tiny tube which connect the sarcoplasmic reticulum and the outer covering as the T tubule. When the T tubule touches the endoplasmic reticulum, that is the sarcoplasmic reticulum, what is stored? Calcium is stored. So it creates a action potential. That is the neural stimuli has become a chemical is released. Now this chemical brings out the further reaction, contraction. That we have to explain, we have to know. At present, what you should know, every muscle cell is a single fiber. It has got outer plasma membrane is called sarcolemma. The cytoplasm is known as sarcoplasm. The sarcoplasm, it has got many nuclei arranged in a row by row beneath the sarcolemma. And the blood vessels are on the periphery. So the capillaries, these are not thick blood vessel, capillaries. So it can, by diffusion, it can get into the cytoplasm. In the cytoplasm, you have got characteristic elaborate endoplasmic reticulum. That is called sarcoplasmic reticulum. Sarco means muscle. And apart from that, in the cytoplasm, there are thin, a microscopic. This itself is a single microscopic fiber. So you cannot see this only with any light microscope. You can see only one muscle. If you mount, you can see. Cockroach. Take the thigh of the cockroach, and thigh part of the cockroach. It has got a broad, like a thigh. It will be a coxa. That's a thigh. You can cut out the chitin, brown cover, and thin fiber, very fresh, nice pinkish muscle fiber will be there. Very little. Yeah, just take it in a forceps. Put it on the slide. Dissection microscope. Whether you stain the color or stain or whatever it is, you just press the car slip. You can see the fiber. You have to tease it. When you should not go with the pen, needle or anything. Just press your car slip on that. After covering, just press it nicely. The fiber will spread. Thin fibers you can see nicely, myofibrils. But the same myofibril, here in human muscle, you cannot see that. Completely that is only under electron microscope. Under electron microscope, you can't see even one big fiber. You can see part and part and part only. But the myofibrils are special protein plate-like structure. Now, these proteins are thin filaments. These myofibrils are arranged neatly. They lie in the cytoplasm in a parallel manner. There are two types of fibers. One is thin and one is thick. So as a result, this thin fiber, the myofibril, has got two kinds of proteins. The proteins are, these fibers are arranged row by row. Each fiber will have a different type. This myofibril. Understood this? Muscle, fascicle, myofiber, cell. This is a single cell. Inside the cell, myofibril, BRIL. This is BRIL, fibril. Now each myofibril, okay? I will write your fascicle. This is the muscle bundle. Muscle bundle. Now this is the this is the sarcolemma. Okay. Right? Now, this myofibril we take, the single myofibril, a single fiber will show a characteristic appearance. That is, each fiber will show a thick. These are the whole thing, each fiber. Okay, now instead of that, please let me go through. A long fiber is there. Now each myofibril will show a light and dark bands. So that is, it will be, part of it will be dark, it will be dark like this. In between, you will have light striations. Light cannot be seen on that. Sorry. These are lights. Each fiber will show a dark and light striations. Now part of it will be dark and then part of it will be light like this. So this is one single myofibril. Like that way, a number of myofibril will be there. Then you'll have the dark area, and this is a single fiber will be like this. One fiber, then next fiber. A number of myofibrils will be there in the cytoplasm. But when they are arranged like this way, then this is next fiber, okay? Now I have to draw another one. I'll draw it closer. Another one is here. So the whole thing will have this area will be light and this area is the dark one. So this is called light band and dark band. Okay, now this full area one is a light band. This is light band. That is what you mean by striated muscles. Now this whole muscle, a single fiber like this way, it has got a number of purely enlarged. A single myofibril will show like that way. A number of myofibril inside the cell. So a number of myofibrils on the fiber packed like that, the whole fiber will appear as striped appearance. So this part, some part will be dark and some part will be light. So this is the light band and dark band. So this whole area will appear darker, and this area will appear darker. This is called dark band, and this area, the next area, will appear light. Now this is a light band in between, and this is a light band. So the entire fiber will have a striped appearance. That's called dark band and light band. Now this is called light band and dark band, but that is now this is the light band, and this area is known as the dark band. See, I have shown a gap in between, but these are all microscopic structure. You can see them another fiber, another fiber, another fiber. You have got another fiber there in between. So the whole place will appear darker. Same and then you have got the light striations that they have. You have got another fiber in between. So one more line, one more fiber, another fiber. Continuation of a single fiber. So the whole stripe, the bundle, the fiber gets a striated appearance because in each and every fiber, you have got many of these myofibrils are arranged parallelly. And each muscle fiber will have a dark area, light area, dark area, and then. So when they are arranged parallelly, exactly all the dark area will one by one and light. So as a whole, the entire fiber will have a stripe like one part will be darker, another one will be lighter, another one will be this is what in the dark band and light band. Do you follow that? Up till this? So don't muddle up. The questions will be there. Objective type as well as statement. Structure alone. Okay? Muscle bundle, bulk of the muscle. It is made up of a number of bundles called fascicle. Fascicles are bundle of each muscle. Bulk of the muscle is made up of bundles. Each bundle is called the fascicle. The fascicles are held together by fascia. Fascia is nothing but a connective tissue. And inside each fascicle, you have got the muscle fiber. The muscle fiber is a single cell. Now each single cell is a muscle fiber. It's called myofiber, not fibril. Myofiber. The myofiber or a single cell has got as a cell, it has got a plasma membrane called sarcolemma. It has got a protoplasm called sarcoplasm. It has got a network inside. Endoplasmic reticulum is broad, that is called Sarcoplasmic Reticulum. It has got many nuclei arranged in row by row beneath the membrane and the cytoplasm. The blood supply on the periphery, on the surface. And in the cytoplasm, apart from the plasmic reticulum, it has got a number of thin filaments. Thin filaments arranged in parallel manner are in the filament. Each filament has got a dark shade, light shade, dark shade, light shade. Now that means what? The single filament is not made up of uniformly the similar type of molecules. You can make out. It may be the place where there is a more darker, maybe the molecules are more. The chemical, naturally, it will be organic molecules. These organic molecules can be a heavier, larger molecules in one area, then another area, lighter molecule, smaller molecule, larger. So the entire myofibril is made up of two different types of molecules, common sense. But these molecules only, we call that as a contractile proteins. They are nothing but protein molecules. So each myofibril is made up of two different protein molecules in a single fibril. So each fibril will have a dark area, light area, dark area. A number of fibrils arranged together in whole cell. The entire fiber will appear like a striped appearance. Now one area of the you pile up a number of fibers like that. Now the whole thing, the whole thing is a. Imagine this is a single fiber. Now outline I have given is a brown. Suppose the whole thing is packed in your muscle fiber. So the entire fiber has got the dark area is known as dark band and light band. The light band is called isotropic band. Dark band is called anisotropic. This is called I band and this is called A band. Now you have to go further into the I band and A band. Okay? Is it clear? Now that let me proceed further then. In that case, see muscle bulk. Muscle bulk of the muscle made up of fascicle, bundled fascicles. Each fascicle is made up of muscle fiber. Now the fiber is a cell, it's a single cell. In each fiber, you got myofibril. The myofibril has got two bands, dark band and light band. Dark and light band. So as a whole, a number of fibrils together, as a whole, is a dark band, then a light band, then a dark band, and this is a dark band, light, then light and dark like this. So as a whole, the entire bundle gets a striated appearance. Dark area, fully dark area here, and this will be light area, dark area. Now light band and dark band. Dark band is called A band, light band is called I band. Now this is isotropic, anisotropic band. Isotropic, and this is called anisotropic band. Anisotropic band. Up to this, okay, we have to proceed. I have no time now, but a class. Now I'll finish with this. Then next class, what we will do, we will complete the contraction of the sliding theory of the muscle, how the muscles contract. That finish the lesson. Then we'll go over to the tissues, so that it will be overlapping with that. So we'll finish both the chapters continuously. So if you finish the muscle tissue, you have to do in animal or organization of the living world. That is the animal tissues. We have done epithelial tissue, connective tissue. Blood, when you come to circulatory system, you should do the blood tissue. Now cartilage, epithelial, connective tissue. So if you finish that, the two lessons will be consecutively. Okay, I'll see you in the next class because I don't want to start with halfway through. This will lead to, we have to know what is this light band and dark band is made up of, and you've got the proteins, how these proteins are arranged, how the contraction of the proteins, the filaments slide on one another, forming the filaments are sliding with one another, how these filaments are arranged, how the striations, though we draw, this is only a band appearance. Okay, the fiber has got the appearance like that, but actually why this appearance comes, how these actin and myosin filaments are made up of, how are they arranged, how they bring about the contraction is the second part of the lesson. Okay, I'll see you in the next class. Thank you. Sir, excuse me. Cut class. Hold on, sir. I can put camera class there. Sorry.